I'd like to move to the next presentation. Professor Yoichiro Sugiyama, please.
Thank you for hearing my presentation. Please show my slide. I'm Yoichiro Sugiyama from the Saga University in Japan. Okay, I'm going to talk a little bit about the possible involvement of oxidative stress in vocal loading induced by brainstem vocalization in guinea pigs. I have no conflict of interest to declare. The vocal fatigue encompasses feelings of tiredness in the voice and a tendency to avoid using the voice, and physical discomfort when talking, and a noticeable improvement in symptoms after getting some rest. It is also linked to the perception of vocal effort and potential of muscular and tissue fatigue in the laryngeal region. And to elucidate this vocal fatigue index is also used with the 19-item questionnaire that helps identify vocal fatigue and characterize the associated complaints. So we think vocal loading is a critical issue for evoking vocal fatigue. Excessive use of the voice can cause damage of the vocal folds and related muscles, potentially leading to the hoarseness and vocal fatigue. This vocal strain can lead to oxidative stress in the vocal fold tissue. However, the use of antioxidants as a treatment can significantly reduce this stress and promote a recovery from the temporary hoarseness. So previous studies have indicated that the antioxidant treatment reduced the oxidative stress of the aged vocal folds. And the other study have suggested that the antioxidant treatment protects the vocal fold from injury and inflammation caused by vocal loading. So we have a clinical question: does vocal loading increase the oxidative stress in vocal-related muscles as well as vocal fold mucosa? To elucidate this issue, the animal study should be conducted to investigate the impact of vocal loading on oxidative stress in vocal-related muscles, which is important to see the vocal-related muscle activity and using animals. So vocalization in mammals has two main pathways. One is a motor cortical pathway, which is used for communication utterances, and the limbic pathway, which is used for emotional utterances, such as crying, mourning, etc., for mating and feeding. This slide shows the two different vocal pathways, limbic pathway and motor cortical pathway. The motor cortical pathway starts with the motor cortex to the reticular formation in the medulla oblongata. And the limbic pathway starts from with the anterior cingulate cortex to the reticular formation in the medulla oblongata, especially in the nucleus ambiguus via periaqueductal gray in the midbrain. So in this study, we focus on this way, limbic pathway. So what animals do we use for experiments such regarding this vocal pathway? Studies controlling vocalization have been performed using monkeys and cats, sometimes bats and frogs, rats and mice, etc. However, the monkeys and cats are suitable animals because they can communicate in ways similar to humans. However, recently in acute experiments, these animals cannot be used. So what is the suitable animals for the kind of experiment? Bats, rats. So we think guinea pigs might be a more suitable option for conducting experiments. So why we use guinea pigs? guinea pig vocalizes use for communication with within the species with audible vocalization, in contrast to the ultrasound vocalization in rat, mice, and bat. Here is a representative vocal pattern of guinea pigs, indicated by the right panel. Support whistle and chatter and chirp sounds. So low whistle sound is a longer call with a distinct rising pitch. The guinea pigs produce vocalization when separated from each other and when anticipating the arrival of food. The purr sound is a very short call, which is used when the seeking close physical contact. So we need to first explore whether the PAG pathway, which is a limbic pathway, in one subject is similar. Our subject, guinea pigs, is similar to that of other mammals. So the aim of the study was to reveal the vocal pathway from the PAG to the medulla in guinea pigs, and to investigate whether guinea pigs can be substituted for the cats and monkeys. So we record the laryngeal, abdominal, diaphragm muscle activities and voice. And vocalization was elicited by an electrical stimulation of the PAG to the lower brainstem systematically. So electrical stimulation of the PAG vocal pathway produce a vocalization with low whistle sound. And this vocal pathway was continuously extended to the ventral medulla, as indicated in the right figure. So they used the vocalization for communication, and they produce sound with human audible range, within a human audible range. And the vocal pathway is similar to that in cats and monkeys. So we think it's a similar mechanism of vocal control that as that of cats and monkeys exists in guinea pigs. So this is substitutable. So this is vocalization-related muscle activity during breathing and vocalization. As you can see this illustration, the vocalization on the right side can be identified by the strong activity of the cricothyroid and abdominal muscle during a PAG stimulation. So electrical stimulation of the PAG can induce repetitive vocalization, and the low whistle sounds are typically evoked by the stimulation. So, you can see the right panel, and you can see here this sound. So when I stimulate PAG electrically, a repetitive vocalization is induced, as you can see here, the sound. So the vocal loading occurs when there is excessive use of muscles involved in vocal production over an extended period, which can lead to hoarseness. So the question is, does excessive strain on the vocal pathway lead to increased activity in all vocal-related muscles? If not, which specific muscles are associated with vocal strength? And the other question is that, do these vocal-related muscles generate reactive oxygen species in response to vocal loading? Unfortunately, we have represented one animal, but we use the guinea pigs and voice and muscle activities of genuine thyropharyngeal, thyrohyoid, cricothyroid, and thyroarytenoid, external oblique, and diaphragm. These voice and muscle activity were recorded, and we stimulate the PAG to induce vocalization at low to high intensities in response with respect to the threshold intensity. The high intensity stimulation was repetitively delivered to induce vocal loading. Fortunately, I have one animal to measure, but we measure the oxidative stress using the formaldehyde. So we investigated changes in vocalization and the activity of vocal-related muscles in response to various intensities of PAG-induced stimuli. And we also examined the oxidative stress of vocal-related muscles, which may be associated with vocal loading. This slide shows the effect of stimulus intensity to induce vocalizations at low intensity, middle intensity, and high intensity. So as you can see, this red arrow and voice. So high intensity stimuli could enhance muscle activity as well as vocal amplitude and specific muscles. Cricothyroid and thyroarytenoid and external oblique muscles. And this slide shows the effect of vocal loading at high intensity stimulation for a long time, such as 30 minutes, 20 minutes. And as you can see, the upper traces of voice. Prolonged loading of the vocal pathway can cause hoarseness. The vocalization was induced even after the 30 minutes, but spectrogram indicated the upper part trace was changed into the hoarseness pattern. So this slide shows the variations between duration and intensity of voice and stimulus intensities. The duration of inspiration and intensity of the voice were significantly correlated with the intensity of the stimuli. And here is the relations between muscle activities and stimulus intensities of TP and GH and ATH muscles, which control the pharyngeal contraction, hyoid movement, and the laryngeal elevation, respectively. However, there is no significant correlation found between these muscle activity and the stimulus intensity. And this slide shows the relation between muscle activity and stimulus intensity of CT, and EO activity, which controls vocal fold movement and expiratory force. As you can see this slide. And the activities of the external oblique muscles showed significant correlations with stimulus intensities. As mentioned before, unfortunately, I have one representative animal in which the oxidative stress of vocal-related muscles was measured. But in this case, external oblique and thyroarytenoid muscles showed an increase in MDA protein levels due to the vocal loading. Additional studies will be necessary to verify this phenomenon. The vocal loading can be applied by delivering high-intensity stimuli to the PAG pathway for a prolonged duration. Intensity of voice and activities of EO, external oblique muscles, are linked to the drive from the central networks producing vocalization. The vocal loading may lead to oxidative stress for TA, thyroarytenoid, and EO muscles. The vocal loading may cause oxidative stress to specific muscles related to vocalization, possibly deteriorating vocal activity. In conclusion and perspectives, we examine the activities of the voice and voice-related muscles during PAG-induced vocalization at various stimulus intensities. The vocal-related activities changed according to the specific stimulus intensities in certain muscles. The vocal loading may induce oxidative stress in the intralaryngeal and expiratory muscles. An antioxidant treatment may help maintain vocal quality after vocal loading by reducing oxidative stress in the laryngeal and respiratory muscles. Thank you for your attention.